Hey guys, it's Mr. Herbst here. I wanted to go ahead and give you a little background information about how this video thing is going to work. I know it's a new process for a lot of you, but basically, I highly recommend taking notes on things that I say. A lot of times the things that I'm going to say are going to be right within the words, right on the PowerPoint here, but occasionally I will say things that are going to be really important, and ultimately I kind of want to train you guys to be really good note takers, and so sometimes I may do a little shout out, say make sure that you write this down. Anyway, our focus today is going to be on biotechnology. We're going to be focusing on Chapter 16, Part 2. I want to take a second to review a little bit recombinant DNA, what we've been focusing on the past couple of weeks. A good example of that was human insulin, where if you recall, we have the human insulin gene, which is here in purple, and we go ahead and we insert that into a plasmid of a bacteria. Plasmid is like a little segment of DNA. They're usually circular. They're like a little segment of DNA that's not actually the bacteria's chromosome. It's just like extra DNA that they have, but that those plasmids are usually really important in the fact that they can help bacteria become like antibiotic resistant and things like that. But anyway, if we go ahead and we take that little gene and we stick it in right there into the plasmid, every time that bacteria is going to reproduce, it's going to reproduce that plasmid, and it's especially useful when we're talking about human insulin, where we can actually go ahead and make human insulin by using bacteria. I want to shift gears and introduce the polymerase chain reaction to you guys. That is abbreviated PCR most typically, so I probably will refer to it as PCR from now on. It's a process that creates a copy of a little segment of DNA, and it requires DNA polymerase, which, if you recall back from a few units ago, DNA polymerase is an enzyme that elongates or extends or lays down new nucleotide bases in DNA. And so ultimately, when we do a polymerase reaction, we are making new little segments of DNA, and we're going to require to have those available nucleotide bases available. So in a few weeks when we do a lab, it'll actually be pretty cool. You will actually be holding in your hand an A, T, G, or C, which makes up all of life's DNA. So here I have a diagram showing the polymerase chain reaction, the PCR reaction. It all starts here with a little segment of DNA. Again, that's not all the DNA available. That's just a small chunk. And we go ahead and we heat it up, where we actually denature the DNA so it separates into its two different strands. And then we lay down some primers, and then we go ahead and we put in our DNA polymerase, which is going to go ahead and extend or lay down new nucleotide bases, and we're going to go ahead and get basically a new segment of DNA forming. And then we go ahead and we heat it up again, denature those, and then we add in more DNA polymerase, and we do that, and ultimately we're able to create many copies of that little target segment of DNA. What are the kind of uses of that? Well, if we think about, we want to kind of figure out where do we evolve from? What's our evolutionary history? And so the nice thing is, if we go ahead and we grab some DNA from a mummy, for example, we can take. We're not going to find very much DNA in there because mummies are 10,000 years old or something like that. We're going to find a little piece of DNA, and we're going to go ahead and do a DNA polymerase chain reaction, and we're going to make lots and lots of DNA from that little chunk, and we can go ahead and kind of compare that to where modern humans are and things like that, and kind of figure out where's our evolutionary tree, where do we go, where do we come from. Another thing we can do with the PCR reaction is DNA fingerprinting, where we actually go ahead and analyze a little segment of DNA and compare it to something else, another segment of DNA. And that can be really useful, especially in like a murder case where we don't really know who committed the murder. So what we'll do is we'll find like a piece of hair on the dead person's clothing or something like that, and we'll go ahead and run that through PCR, make lots and lots of copies of that DNA, so we have plenty to go ahead and compare it to, and then we'll go ahead and test it against a few known criminals and kind of figure out maybe who did it. And it's also really useful in paternity tests, like if we want to find out who the mother or the father of a certain baby was, we go ahead and we do a PCR reaction and make plenty of copies of that DNA, kind of compare it to other known segments of DNA. How do we compare it? Well, we use this thing called gel electrophoresis, which looks like this, and we'll actually go ahead and use something that looks like this in a couple weeks here, where we actually load in, right in here, we would load in little segments of DNA, and eventually you would run that, let it go for a couple hours, and we'll get this pattern that looks like this, where we have all these little segments of DNA, and based on how big the segment of DNA is how far it moved. And again, I've been talking about this recently. Smaller segments of DNA will move farther. So if we say up here is the top, and we load all these little segments of DNA at the top here, that would mean that this segment of DNA right here would be actually the smallest segment of DNA, because again, it can move the farthest. It's much easier to push a small person than it is to push a big person. So big chunks of DNA don't really move very much. And I want to talk about a few examples of transgenic organisms. For one, bacteria, where we actually can use recombinant DNA and grow bacteria in large vats like this. And we've talked about this before. The uses of having transgenic bacteria is, for one, making human insulin for us, but also there's a whole lot of other uses. We can make bacteria almost make any protein for us. Here's a few examples. Human growth hormone, vaccines, certain oil-eating bacteria, so we can actually alter the way that what bacteria eats, and so we can actually change some bacteria to eat oil, so in case there was an oil spill, we can now use bacteria to kind of clean it up. We also, we also use GMOs for plants. You probably heard about that commonly in the grocery store, where we actually take little genes and implant them into immature plant embryos. And a couple good examples of that are this thing called a pomato, which it's kind of funny to say, but if you take a look here, those things down there are actually potatoes growing in the ground, and up here we actually have tomatoes growing on the top. It's actually kind of interesting, but the implications or how the usefulness of that should be screaming at you. Now, if you think about all the land just used for growing potatoes and all the land just for used growing for tomatoes, what if we could grow both on the same amount of plot of land? I mean, now we're talking about efficiency. Another example of transgenic plants are BT corn, and that's a type of corn that you've probably eaten it at least one time in your life, but BT corn actually has the ability to produce what they call the BT toxin, which actually kills little insects that will eat that try to eat that corn. So it's like the corn actually is producing its own pesticide. Pretty cool. We also do transgenic operations on animals, where we actually insert genes into animals when they're little eggs or tiny little embryos. And some examples of that, one of them really is kind of cool actually, where we actually put genes for producing spider silk proteins into goats. Now, what I mean by spider silk is spider webs. Spider webs are made by proteins that spiders produce. Now, what is the benefit? Well, actually, spider webs are one of the strongest materials known to man. Now, you may look at one spider strand and you can rip it apart, right? But if you had like a whole rope made out of spider silk, it would be stronger than a steel cable. So actually, spider silk in the future has a lot of different potential to be one of the best building materials possible. And it's difficult to harvest, right? Because we would need a lot of spiders. Now, the problem is spiders are actually very territorial, and they tend to kill one another that are nearby. So what we've done is isolate the little protein for making spider silk, and we stuck it in goats. And now when goats go ahead and they make their milk, that little protein is actually produced in their milk. We can isolate the protein, and thus we can artificially make spider silk. Really cool thing that we're actually doing with transgenic animals. I want to shift gears here and talk about for a second about genomics. There's another topic we're going to learn about in this unit, and that is the study of genomes. And if you're not familiar with what genome is, a genome is all of the genes in your entire body. So if I was to want to study all of the genes in my body, that would be my genome. And so genomics is the study of genomes. We've done this where you may have heard of the Human Genome Project, where it was actually finished in 2003, and at the time they discovered that in the human body there were about 25,000 known genes. And since that time, we've actually discovered that it might even be less than that. But the magic number right now is around 20,000. So actually, since the time of the Human Genome Project, we've discovered that it actually is a little bit less. We also go ahead and we like to compare our genome to, say, chimpanzees or some other form of very similar organism. And we've also been working on this HapMap project, where we go ahead and we compare our genome, my genome, to another human being, and really ultimately find out those little differences. And what the future is going to hold is actually pretty cool. We're going to have these things called DNA chips sometime in the future, where actually you in the palm of your hand you'll be actually able to hold a little chip that'll show you if you're susceptible to certain diseases or not. And the question I kind of want to ask you guys is, who's going to have access to this information? Anyway, guys, that concludes biotechnology. This is Mr. Herbst. I'm signing off, folks. Y'all have a nice day.